Thank you. Thank you to the organizers for inviting me. So there's some advantages and disadvantages of being on the last day. But one of the advantages is that you can maybe calibrate a little better to the audience. So I decided to ditch what I was originally going to talk about and maybe talk about some things that are not as new, but presented in a way which might be more interesting for the people that are computational, which there are a lot of those here. So I think that we've all seen examples of how useful modeling can be, and we know how sort of brain-like networks are useful for all kinds of machines doing intelligent things. But I think that we all agree that even the most intelligent systems today are not really as intelligent as the brain, and that there are many things that are easy for us to do that are really hard for artificial networks. And the question is, what are they missing? And maybe what they're missing is some of the features that our brain has that are not normally integrated into these types of networks. And so the first thing is that many of these models, or actually pretty much all of them, use a generic neuron. And there really is no such thing as a generic neuron. Neurons are very diverse in their shape and their size, and they're not connected in a way that is uniform. In addition to that, every neuron has a different organization of inputs onto it. And in that sense, I mean not just valence, excitatory, and inhibitory, but inputs from different sources that convey different types of functional information. And these, of course, are distributed differently and allocated differently onto different cells. And the third thing is that brain circuits are plastic, and they constantly undergo a use-dependent optimization. And so I wanted to actually go through this very simplistic model of a cortical kind of microcircuit and suggest to you that this plasticity, even at the level of individual synapses, can make a difference not just to the computations within the neuron, but to the local network. So for example, here we have a generic pyramidal neuron from layer 2-3 that's receiving both excitatory and inhibitory inputs. The excitatory inputs, again, simplistically, can be divided into feed-forward inputs from the thalamus or top-down inputs from higher cortical areas. And the inhibitory innervation can be divided according to the part of the neuron that is being targeted, where we have somatostatin neurons targeting dendrites, PV neurons targeting the soma and proximal dendrite, and then and also some that innervate the axon initial segment, and of course those can all have different impacts on the neuron. So let's say you have this stable circuit in the cortex, and then what you do is you just remove this one synapse that's receiving top-down information, and that and the branch of a nearby axon will now innervate another synapse at a very close location. And then you remove one of the inhibitory synapses from a nearby spine or from the dendrite, and effectively, by making maybe 4 synaptic changes, you've kind of reweighed the local inputs to favor the bottom-up versus the top-down information. So, so I wanted to introduce this phrase that Idan uses that I've adopted because I like it, is that there's such a thing as strategic placement of synapses, and that strategic removal or addition of synapses can effectively rewire circuits without a lot of large-scale anatomical reorganization. And the question is, can we integrate this type of information into models that would be more realistic? And so the idea is to introduce accurate synaptic mapping information into models to make them less generic. So the way that we approached this was we developed a method to do wholesale synaptic labeling in vivo. So this is the only molecular biology slide, I promise. So it's a 3-color system. And in a minute, you'll understand why these are upside down, where we use YFP, which yellow fluorescent protein, as a cell fill. We have Gefrin, which is a scaffolding protein that is common to all inhibitory synapses, fused to teal, which is a blue fluorescent protein. And then we have PSD95, which is supposed to be common to all excitatory synapses, and also a receptor scaffold, and it's fused to M-cherry. And these 3 fluorophores are in different plasmids, and they're in these dio cassettes, double inverted cassettes, where they don't express except in the presence of Cre. And so what we do is we put all 3 plasmids together at high concentrations so that cells will get all 3 colors, and then we put a limiting amount of Cre, and only the cells that get the Cre will actually, will express all 3 plasmids. So many cells will contain all the plasmids, but only a few will express the Cre, and we'll get the sparse labeling that we need for imaging. And these plasmids are electroporated in utero into the ventricle at E15, which is when the layer 2-3 pyramidal neurons are being born at the ventricle. And the plasmids take up the DNA, and they then differentiate and migrate to their correct location, carrying all the colors with them. And these, after these labeled pups are born, when they're about 6 weeks old, we replace a part of the skull with a cranial window that allows us optical access to the neurons. After they recover from the surgeries, we use intrinsic signal imaging to map binocular visual cortex, and we pick cells specifically in this region. And then we monitor the same cells under a two-photon microscope every 24 hours for, in this case, 8 days at 24-hour intervals. So this is what one of those triple-labeled neurons look like. You can see the soma and axon initial segment receive really heavy inhibitory innervation. That's why they're so bright. And now when this kind of moves towards you, hopefully you can see these little dots, where every yellow dot is an excitatory synapse. You can see they stick out a little bit. They're on the spines. And then you can see these blue dots, which are the inhibitory synapses. And you can see them even when you go down to the deepest basal dendrites. You can score every synapse. Here you see the axon with the boutons kind of as we pop out. So we have a good view of the cell and all the synapses on this cell, both excitatory and inhibitory. And for scoring, we would zoom in, and this is what it would look like. So you see here the cell fill. You see all the spines on this dendrite. You see in yellow the PSD95. In this case, every spine has a excitatory synapse. And here you have the inhibitory synapses in blue, and you can see these 2 inhibitory synapses on the dendritic shaft, which is what we were expecting. But we also found a large number of inhibitory synapses on dendritic spines together with an excitatory synapse. And so what do we have here? We have a view of the entire cell. We have the spines. So this would be equivalent to what you would see if you were doing a Golgi stain, for example. So that's the kind of information that we might have, in a static view of neurons. But then we also have the location of the inhibitory synapses, which we initially expected to be on the shafts. And then we saw that there was this population of inhibitory spine synapses. But because, and these had been anecdotally described by EM, but sort of always as if it was some odd thing to see a spine that had two synapses. But because here we have a view of the entire cell, we could actually say that these are the inhibitory, this is color matched to here, that the inhibitory spine synapses are actually a third of the entire inhibitory synaptic population. So not an insignificant fraction. So that suggests that the inhibitory population is maybe not uniform in terms of their potential role in dendritic integration. The other surprise that we had is when we looked at the PSD95 labeling is that not every spine had PSD95 labeling. And this is, the reason this surprised us is that if you do EM on adult tissue like this, pretty much every spine has a synapse structurally. So that suggested that at a molecular level, spines that look the same, either when you do a cell fill or when you do EM, are not necessarily the same. And so that suggested that there aren't just two potential inhibitory synaptic populations, but also potentially three spine populations, because we have these spines that don't have PSD95. We have the spines that have just PSD95, which we expected actually everyone to be. But then we also have this population of about 15% of spines that are duly innervated by both an excitatory and inhibitory synapse. So even by just doing kind of a one-shot imaging at this level with having the synaptic labeling, kind of allows us to add potentially one layer to the type of information that we might have by doing some static anatomical stain like a Golgi or a cell fill. And so this is the kind of modeling that we would like to do, and this was done by Michael Doron and Idan Segev's lab here as the start of our collaboration. And he actually didn't use our data, but data that was available from cell fills and with synaptic labels. And this sort of initial model was looking at whether the way the synapses are distributed would potentially affect the way inhibition impacts the excitatory inputs. And so, so the first thing that Michael noticed is that the inhibitory synapses are actually seem to be distributed a lot more evenly than the excitatory synapses, and more evenly than one would expect if they were just randomly sprinkled over the cell. And so what he did was he modeled how that would impact potentially excitatory NMDA spikes in the dendrites. And so this is a model of a cylindrical dendrite that is receiving, I don't remember, I think he did 30 excitatory inputs and also receiving inhibitory innervation. And he asked whether it made a difference if the inhibitory synapses were evenly distributed versus randomly distributed. And what he saw is that if he had even distribution of the inhibitory synapses, sorry, let's go. If you have excitation of all these excitatory inputs, if they were all active, I think there were about 30 excitatory synapses that would generate an NMDA spike in the dendrite. If the inhibitory synapses were evenly distributed, they would potentially totally attenuate the NMDA spike, while if they were randomly distributed, they would maybe attenuate it, but would not really be sufficient to block it. And so the fact that we were seeing inhibitory synapses evenly distributed meant that they could have potentially higher impact on whether NMDA spikes would be allowed to go through or not. So this is sort of like the very minimal analysis that you could do on the static picture. But the advantage of having the imaging data where we have multiple sessions is that we could actually ask how the network is changing, how the inputs onto a given cell are changing over time. And so this plasticity is something that has not really been integrated into models yet, and I want to talk about it from several different perspectives. And the first perspective is the excitatory perspective. And so looking at the excitatory inputs, which is basically the spine. So we've had several talks here where people have used spine dynamics as a measure of plasticity. And what we see here is that actually not all spines are equal. And that spines are, first of all, unequal in terms of their synaptic distribution, but also this unequal synaptic distribution reflects a different dynamic behavior. And so if you look at the spines without PSD95, you can see that even though they really account for only about 15% of the spine population, they account for 80% of the dynamic. So they're, sorry? Gain or loss within a 24-hour interval. Yeah. Yeah, so I'm not actually talking about size changes here, although the data is there. And I'm talking about a dynamic in the sense that a gain or loss reflects a loss of input and a different connectivity for that neuron within the network. So the perspective is not just in terms of how the neuron is processing information, but also what kind of information it receives at any given time. Okay, so these spines without the PSD95 are extremely dynamic. The ones with the PSD95 are occasionally dynamic, but a lot less than the ones without the PSD95. And then these spines with both PSD95 and Geffen are extremely stable. We have very, very few examples in hundreds and hundreds of spines that we've seen where we've seen a spine like this change. So when you're looking at the spines actually without a synaptic label, you can't really distinguish between these categories that are molecularly different. Okay, so let's look at what these three different categories mean. What does that look like? What do the dynamics look like? And so this is the kind of dynamics that spines without PSD95 exhibit. So here you see an imaging sequence of a dendritic branch. And you see here is one spine that has PSD95 on it. And you see this spine is stable and the PSD95 on it is stable throughout the whole set. So this kind of shows that the imaging quality for all of these is equivalent and comparable. And what you see here is that this region, which has no spine, on day 4, you have a very small spine. You can see it, it's kind of dim on the, hopefully you can see it. And it pops in for 1 day. And then on the next session, 24-hour later, it goes away. And then on the next day, you see another spine pop in, not in the same location, close by, and it pops in again for 24 hours, and it disappears. So this is what those rapid dynamics, or this is what those dynamics of the spines without PSD95 looks like. And so most of the dynamics are of this type of the spines. Then we have the spines with PSD95, and this happens less frequently, but the way that we would see that is that you still see the spines kind of pop in. Here you have two spines. Again, you have a stable spine here that's stable throughout all of these sessions, and you have here two new spines pop in, and then they stay. And here we can begin to see the PSD95 on the second day that this spine is here. And then by the third day, we can see both spines have PSD95. And then once they gain the PSD95, they become stable. So these kind of changes where you have a spine emerge, recruit PSD95, and stabilize are the kind of changes that really reflect a new connection for this neuron within the circuit. So these different dynamics are really reflecting a different kind of connectivity where the spines without PSD95, the connectivity, potentially there is a synapse there, which we can visualize by EM, but it's a transient synapse. While the ones with PSD95 are reflecting changes to the circuit, and their addition and loss reflect kind of a long-term reorganization. But most of the changes are actually of this type and not of this type. So what would be the logic of this type of dynamics? Well, the logic would be that potentially most of these transient events are just sampling potential partners and testing out new locations, and only in about 10% of cases those contacts are selected for stabilization. And so, so one of the questions is, what is really determining which of these transient contacts is being stabilized? And we all know that activity is a driving force for plasticity. So the question is, kind of at what stage does experience or, oh, sorry. So actually, I already said this. So basically, most of these are trying out different locations. And just this is to kind of remind the people who are doing spine dynamics that since most of these spine dynamics don't result in stable circuit changes, then the dynamic spine counting may not really reflect the actual plasticity. And so then the question is what reflects the actual plasticity, and what reflects the actual plasticity is which synapses get stabilized, and that's where experience plays a role. And the question is at what aspect of the synapse formation is activity influencing. And so the way that we can look at that is, since we do map these cells into visual cortex, these are cells that receive visual input, we can manipulate the visual experience and ask, does it influence the spine dynamics? And so in this particular experiment, it's a very similar experimental paradigm to what I showed you before, except we did longer imaging intervals, because it turns out that the excitatory changes tend to be relatively slow. And when we're only looking at excitatory, there's no point in doing daily intervals. And so what we did is we did 2-week intervals. And what we did is after the first 2 weeks, so we had a baseline 2-week interval, then we put the animals in the dark for 2 weeks, and then we took them out again for another 2 weeks, and we had imaging intervals before and after the dark. And then we asked in the different spine categories who's impacted by this. And what we have here is the number of spines gained and lost at each one of these intervals divided by category. So what you can see here is these are the PSD95 minus spines. You can see the scales are different because, of course, these spines are much more dynamic. But you can see that these spines are dynamic to the same extent whether the animals are in the light or the dark. So the emergence, the gain, and the loss of spines, these transient dynamics, are entirely uninfluenced by the visual experience. On the other hand, the category that seems to be most influenced are the PSD95 positive spines. So that means that those events where you are actually gaining a PSD95 and keeping a synapse, that's a gain, or where you're losing the PSD95 and losing a synapse are the kind of events that are diminished in the absence of experience. And so it seems that the aspect of excitatory synapse formation that is influenced by experience is actually not the spine emergence, but the decision which spines get stabilized. This is just kind of another little interesting point is that the dually innervated spines whose baseline is extremely stable also are stable when you manipulate experience, which means that things that tend to be dynamic are things that are dynamic anyway, that something that tends to be stable is not influenced. So, so my suggestion would be that actually since this category is the only one that is influenced by experience, then really the measure of PSD95's positive spine dynamics is a much better reflection of excitatory circuit plasticity than net spine dynamics, where you're not actually looking at the fraction of the circuit that is long-term and is impacted by experience. Okay, so now I wanted to switch to the inhibitory perspective. And here we'll look at the inhibitory synapse dynamics broken down by category, which is either shaft or spine. And what you can see first by this scale bar that overall the inhibitory dynamics are much, much higher than the excitatory dynamics. So it seems the inhibitory circuitry is more plastic than the excitatory one in terms of changing connectivity. And the other thing is that between these two categories, the inhibitory synapses on the spines are particularly dynamic. And I think it's pretty remarkable that if we image over 8 days, 60% of those inhibitory synapses on the spines will undergo at least one dynamic change. So this is really pretty drastic. So what does this look like? So this is a time series where this is all 3 channels. This is just the Geffen channel. And again, you can see here, there's a stable shaft synapse that is there across all the imaging sessions. These are the outlines of the spines. And you can, if you pay attention to this spine here, you see it has no inhibitory synapse, none. Pops in for 1 day, goes away, pops in again for 1 day, then goes away again. Then here you have another example. Here you have on this spine an inhibitory synapse that is there, all the way up to here. Then it goes away for 1 day, and then it comes back for 1 day, and then it goes away again. There's another one near it. This looks a little different because it's in a Z projecting spine, so the angle is not as clear. But you can see here that it's not here on the first day, and then it goes away, and then it shows up, goes away again, and then it's there, and then it goes away again. So you see that these inhibitors, this is an interesting example because it shows that each of these inhibitory synapses are dynamic, but even though they're so close to each other, they're independent. They don't show exactly the same behavior on each day. But also, if you notice, there's a difference in how these synapses come and go from the spines, where with the spines, what we saw is that most of the changes had to do with sort of popping out at different locations. And when you're sampling partners, it's good to try out different locations. And only a few of those are stabilized. In the case of the inhibitory synapses, what we're actually seeing is that the inhibitory synapses are coming and going in the same location. So that's not about sampling partners. And what we're suggesting is that this is about reversible modulation at specific sites on the dendrite. So, so potentially the purpose of inhibitory remodeling is different from the purpose of excitatory remodeling, where I guess one perspective would be that it's all about the excitatory synapses, where they get to pick which are the stable contacts, and then the purpose of the inhibitory remodeling is to do a different type of picking of which excitatory synapses are the ones that participate in the circuit. If we were to go back to this initial modeling that Michael did, now we have the opportunity to layer in some additional type of information, which is, for example, now that we know what impact, or we would know what impact specific strategic placement of inhibitory synapses have, then we have the opportunity to ask, if any one of these were removed, would it matter or not? Does the location of where you remove the inhibitory synapse matters? How many do you need to remove in order to have an impact, and in what type of sort of organization would this removal happen? So we have kind of this additional type of information. Now, what about the relationship between these different types of dynamics of the excitatory and the inhibitory network? So the first thing is the question of whether, when you have a dynamic event of an inhibitory synapse, how likely is it to have a nearby dynamic event for an excitatory synapse? And so when we did this very simplistic nearest neighbor analysis, what you can see here is that you have, if you have a dynamic inhibitory synapse, the likelihood that you would have a dynamic inhibitory synapse, the likelihood that you would have a dynamic spine would be much higher. Sorry, which one is it? Yeah, so this would be that, and this would be the converse of that. If you have a dynamic spine, the likelihood to have the inhibitory synapse. But this pairing doesn't work for excitatory, and it doesn't work for inhibitory. So it's a specific pairing of an excitatory and an inhibitory remodeling that, that we see. And if we actually ask what's the distance for this pairing, we can see that if you have a dynamic inhibitory synapse, more than 50% of them have a nearby dynamic spine, and then another almost 20% have a nearby, two nearby dynamic spines. So a large fraction of these, if you add 60, almost 80% of dynamic spines are coordinated, dynamic inhibitory synapses are coordinated with a dynamic change in an excitatory contact, potentially. So, and the second thing that we could look at, aside from this local clustering, is kind of looking at each one of the dynamic categories and to break them down for each type of excitatory or inhibitory input. So, so for example, in the case of spines, we see these changes where, and inhibitory synapses, we see these changes that are transient, which means the synapse popping in just for one session and then going away, or the spine popping in and going away. So this is something that we see in both cases. We also see these one-time dynamic events where we see a synapse added or a spine added, and then stabilized or lost and then remain gone. And this, again, is common to both the excitatory and the inhibitory type of dynamics. But then we also see this recurrent behavior, which is really specific to inhibitory synapses, and which is synapses that pop in and out at the same location. And let's look at these categories in relation to all the synapse types, where here we have all the excitatory, here we have the inhibitory. And here we have the spines without the PSD95, and then we also have the PSD95 on stable spines, and the PSD95 on, which are just PSD95 positive, and these are the PSD95s on duly innervated spines. And again, you see that these duly innervated spines are not just stable themselves, but the PSD95 on these spines never comes or goes. And then you see here the inhibitory synapses, which overall are much more dynamic, but also that they have this large recurrent category. And I have to say that this is a conservative estimate of the recurrent category, because for us to score something as recurrent, it has to pop in relatively early in the series to be able to go away and come back. So potentially some of these things that we're seeing as transient or one time because they're coming in later in the series are, if we waited longer, might be of this recurrent type. So the question is, how do these relate to each other? And I think that the best way to look at that is actually to look at these two, because these are two synapses that are on the same spine, so very close to each other. And what you can see is that these two synapses that are on the same spine represent the two extremes for each category, where these excitatory synapses are the most stable excitatory synapses, and they're paired with the most dynamic inhibitory synapses. And so, so what we think this suggests is that maybe when you have an excitatory synapse that is super stable, but you don't always want it participating in this circuit, one way that you could take it offline would be to attach an inhibitory synapse right next to it, and in that way take it temporarily offline, but not really dismantle it. And so potentially these type of dynamics are really not independent of each other, but related. And this actually can take us out of the thinking about it just at the level of the neuron itself and its integration, but in relation to the local circuit and connectivity, because it means that not all contacts onto this neuron are being treated equally in terms of their stability. So it suggests that there are actually contacts that are potentially so important that you're not allowed to remove them at all, ever. And then you could still take them on offline through the use of the inhibitory synapse. But the question is, wrong spelling, but what is it about these excitatory synapses that make them privileged? What types of contact in the brain are really hardwired, essentially, while others are plastic? And I think this is also an important aspect where it means that not all parts of the circuit are necessarily equally plastic, and there are parts of the circuit that are stable and some that are not. And so these are the type of new experiments that we're doing now, which is actually to try to ask whether we can identify who the inputs are onto specific synapses that have a different dynamic behavior. And so my favorite idea going into these experiments was that these stable excitatory contacts are potentially the feed-forward thalamic inputs, and that maybe the cortical inputs are the ones that are more plastic. And so, so this is the first generation of experiments that we're trying out now, where we're using actually one of the colors on the presynaptic afferents in order to mark where they come in and relate that to the dynamic of the postsynaptic side. And so this is a combination of the molecular labeling strategy that I showed before with a genetic labeling. And so what we do is we have a mouse, an olig-3 Cre, which is a thalamus-specific Cre. And we cross this thalamus-specific Cre to a mouse that is a lock-stop-lock synaptophysin TD tomato, which marks presynaptic boutons. And so this, the progeny of this cross should have all their boutons labeled in red. And then when this mouse is pregnant, we do the in utero electroporation as before. We put the windows on, and so this is the idea that we would have the cell fill through the YFP. We would have the synaptic label through the PSD95 teal. Here, this is an excitatory line, so we're using the excitatory postsynaptic label. Because this is in a Cre background, these are now flip constructs. And then we would have in red, the boutons of the thalamic afferents, and what we would be looking for is this overlap of a thalamic bouton with a PSD95 positive spine, and ask, are these really the most stable contacts? So this is what, in principle, these experiments are working, where we have here, we have the cell fill, we have the PSD95 label here in white, and here you see the boutons. And so this is an example of a bouton where we have an overlap of the PSD95 with the synaptophysin TD tomato, and then here we have two spines that have the PSD95, but they don't have the presynaptic afferents. So those would be cortical contacts. So I won't tell you the answer yet because I don't know it, but hopefully we'll be able to answer those types of questions too. So, so the layer 3 of this type of modeling would be to integrate the different afferent sources with the location and dynamic behavior of the synapses. So to summarize, what have we learned from in vivo imaging of spines and synapses? And one is that there's actually 3 types of spines that differ in their dynamics, ranging from hardwired to ones that are partner sampling. That the most dynamic spines are the ones that lack PSD95, and that the logic of that seems to be about maximizing site testing, and infrequently leads to remodeling. And that inhibitory synapses, many of them show recurrent dynamics at stable sites, and that most dynamic inhibitory synapses are on duly innervated spines situated near a hardwired excitatory synapse, and that potentially this means that the inhibitory dynamics are reversibly gating the excitatory circuits. And finally, these are the people that have done the work I talked about. So Jerry Chen was the one who started the inhibitory, a former student, now has his own lab, who started the inhibitory synaptic labeling. Katie and Kalen integrated the third excitatory label into the system. Jay was the one who did the activity-dependent remodeling and work that I mostly didn't talk about. And Aigul is doing the thalamic afferent labeling. And all the imaging in my lab is done in collaboration with Peter So's lab in the Department of Mechanical Engineering. He's the optics expert and the reason that our imaging looks so great. And Jaewon is his student that built our 3-color system. And Michael started and will be continuing to do the modeling on our data, and he's in Idan's lab here. And thank you for your attention.